I'm Wendy Herzog, science and peptide enthusiast. In this episode of Exploration Science, I met up with Dominik Sarma and Oliver Reimann, co-founders of Belyntic. They walk through their peptide easy clean or PEC technology and its applications from discovery through manufacturing of peptides. They also talk about the process of starting a company together. Again, thank you both for doing this. It's going to be fun. I love the story of Belyntic. I think I put a LinkedIn post about the webinar that one of your scientific co-founders, Robert Zitterbart, put out that was with Bochem, right? Talking about the story of Belyntic. But maybe you guys could just quickly talk about how you know each other.
Great. Yeah, so Dominik, Robert, and I actually started studying chemistry together here in Berlin, in Germany, at the Humboldt University. It's already quite some years ago. It was, I think, 2005, and actually started as a friends and study group, and then after some study years, I think the ways had kind of separated from one another, and one went on to, I think, Dominik, you went to Spain for a while. Robert did his diploma thesis in the US, and I went to another institute later on and did my PhD also in the Leibniz Institute. And then after a while, all of us came back together and did kind of a brainstorm on where our competences that we were working on also in the PhD time had a good overlay. And this was, in principle, what we came up with was that Robert and I used to work on peptides, and especially new chemical linker entities to connect peptides with other molecules or to even purify them. And Dominik was working on, let's say, had a lot of experience in the field of material-based chemistry. So this is in principle where the ideas merged, and we came up with the idea to try something by our own. And in the end, with this idea, we applied for a program in Germany to drive out innovation from the university towards the market. And with only, let's say, a couple of experiments directly in hand, we got this grant, which allowed us to develop Belyntic for 18 months until the point where we incorporated the company in 2018.
Wow, yeah, that's awesome. Let me ask a question. Oh, sorry, go ahead, Dominik.
I just wanted to add that before we actually called ourselves EnviroPep.
Oh, okay.
The precursor of Belyntic was EnviroPep, but when we founded the company in 2018, we changed the name just to have like a more generic name that's not so much connected to peptides. I mean, we're doing peptides mainly, but the linker has many possible applications. So we were trying to find a word that describes in a little bit more broad sense of what we do. And so Belyntic is like Berlin linker technologies.
I was wondering. All right. Nice. So I have a couple questions around that. One is, how did you get together and formulate that name? Was that another beer session, or was that I'm just curious to know how that creative process worked.
The idea and then the name?
Mm-hmm. Well, the name. So I was really interested in the name.
The name. Yeah, actually, so we had this name EnviroPep. Yeah. And we were thinking, okay, it will always connect us to peptides, right? It has this part of peptides in it. And we wanted to have just a general, nice sounding name. Mm-hmm. And so we decided first to change the name. So that was the first initiative. And then we came up with, I think Andreas, one of the co-founders, he managed the workshop. I think it was self-organized, and he did some games where we started writing names, and then we passed it to our neighbor, and then we tried to extend the name, and then we ended up with a list of 10 names after we ran through those games more or less. And then we did, of course, a cross-check if these are available, like if the domains are available, if there are any conflicts with other trade names. And so I think Belyntic wasn't in the top 3. I can't remember exactly. It was not on the top list. And I'm not 100% sure what was the final decision making event that led us to take Belyntic. But we thought it's nice because we have Berlin, it's BE, linker. It's actually in Berlin. It's with an Y, which is just some style thing, and also technology. We thought, okay, everyone is tech in the end, so we just change it a little bit to TIC, tick, and so it's Berlin Linker Technologies, but in a very
and you won't believe how many domains are already taken from names that you think that impossible that someone came up with this before.
Yeah.
Well, and I think we're pretty happy with the name, right? So yeah. I think it actually does exactly what we wanted to do. It's just a name, and it sounds good as a name, and it doesn't connect us to some specific market or field of application. It's a broad name, and it will open up new doors for us maybe in the future as well.
I think so. I think, having a unique name that's also, when I first saw it, I was like, wait, how do I say it? How, I had to think a little bit about it. So I agree. I think it's great. Honestly, it's a good, a way to have a brand. So it's very nice. Tell me a little bit about Belyntic, so what, your technology and sort of the business model that you have.
Yeah, so what we have developed is a new linker-based method to, let's say, purify peptides. That's the main idea. So in general, the status quo and the most well-established technology to purify peptides is by chromatography means, usually HPLC, but also maybe flash chromatography. And so, to be honest, in our time when we worked with peptides, we had a lot of experience with these kind of chromatography devices, and we always thought that it would be really great to have alternatives at hand that would allow for different kind of approaches. For instance, to work more rapidly, like to have a very rapid parallel working technology at hand. And the way to accomplish this is very difficult by chromatography, where you always have you run a flow of a gradient of solvents through a column and try to separate the compounds. But this is hardly possible in high parallelity. So that's where we came up with the concept of using chemical-based methods, which allows us to work in reactors. And so this is, in principle, what the main idea was, to offer new ways that are totally different to what's out there to purify peptides in the end. And nowadays, we also initiate a lot of projects where some other benefits become also obvious of our technology, where, for instance, the complexity of peptides nowadays of modern drugs becomes quite difficult for manufacturing. Sometimes you have some kind of conjugates to, let's say, PEG groups or lipidations, sometimes multiple unnatural amino acids that really complicate the manufacturing and purification process. And one of the nice benefits of our technology is that it's a one-size-fits-all method. So it's rapid. I just do the scheme that I know for every single peptide that's out there. I just apply it and I get a certain results out of it, and I don't have to cope with solubility of my probe or other problems that or pooling and fractioning of like it's usual after HPLC. So it's like a one-size-fits-all method based on chemistry. So that's like the key.
And can you walk through that process of the actual peptide easy clean or PEC technology? You can talk a little bit about the chemistry and the steps. And I will say, before you get into that, the website has fantastic videos. You have a lot of really, really great information on the website. So anyone who wants to go actually see like nice cartoons of what you're about to describe chemically, it's on the website. So yeah, just maybe walk through that process.
The principle is that in solid phase peptide synthesis, one amino acid is attached after the other in a building block fashion. And we are able to block unreacted couplings. So if a coupling is incomplete, instead of elongating an incomplete, like a chain where one amino acid is missing, one could block this particular site at the solid phase resin, so that in the end, only the full length peptide would have available N-terminus to couple the linker molecule to the peptide. So that in the end, after the peptide is cleaved from the solid phase resin, you have a crude material where you have all kind of so-called capped or N-terminally acetylated truncations inside, and the full-length sequence equipped with a linker molecule at the N-terminus. And once you have this crude material, we can solubilize the crude, and for this we can take organic solvents that are really well in solubilizing even very hydrophobic peptides, such as HFIP, hexafluoroisopropanol, which is a really good solubilizer, or DMSO, for instance. Solubilize the peptides and put them on in a reactor in which we have agarose beads, which sit there. They are chemically modified with functional groups, aldehydes, and our linker molecule has a site that selectively reacts with these aldehyde groups on the resin. So once we throw the peptide linker conjugate on the solid support, it binds covalently, and this is a big difference to chromatography, where the peptide would interact with the solid stationary phase. So we bind the peptide covalently to the solid support, which allows us to wash out all kinds of acetylated truncations or other synthetic dirt. And finally, cleave the linker molecule in a traceless fashion with very mild stimulus. It's a reductively cleavable linker. So we're using a very common chemical in the lab of peptide chemists called DTT. And so this will then, in the end, cleave the linker molecule and release the peptide very mildly after this washing process that has been carried out. And by this, we can, without the need of machines, work in regular SPPS reactors to generate purified peptides.
So a couple questions around that then are, where do you see this technology fitting in the scheme of from discovery to manufacturing?
I think that one of the nice parts of our technology, it actually fits into each step of the value chain. Because first, as Oliver mentioned, we can run the purification in parallel. So we use, for example, 96 well plates that can be handled in liquid handling devices. So we can do this in an automated fashion and create very fast libraries of purified peptides. So let's say the moment a drug developer has maybe 200, 300 potential leads in hand, which is a common number, I would say, after first screening program, we can run them all. Put it like this. So one could really work with the whole pool of potential peptide leads. One does not have to stick to 10 or 20, because otherwise it will get very, very lots of workload with chromatography. So this is the first step during the, let's say, drug development chain where we could work in parallel. Then one step before the cleavage of the linker, we have a situation in which the unprotected peptide is loaded on the agarose covalently. And that's it's a sweet spot, so to say, because to this moment you can run modifications. So you have not only a purification method, but optional modification step. At this point, we can do lipidations, pegylations, dye couplings, all this simple chemistry, but also the difficult ones, cyclizations, disulfide formation. So the whole variation of possible modifications that are needed, and that's the second step, during the lead optimization step. So we work in parallel. We can integrate a modification to speed up the whole initial hit to lead and lead optimization cycle, because it's all coming from one technology. You basically need the PEG linker and to run it, and certain machines and devices. It depends on how you want to set it up in the end. And then we call this design for manufacturing, because it's, let's say, our intention to make developers work with the best possible drug. That is, regardless of the complexity, we can generate with our technology. We can also scale it up. And you will still benefit from the features of PEG, as it saves lots of organic solvents. It's easily scalable in available SPPS reactors. So you can basically use the PEG technology along the whole value chain, speed up the process initially, work with the best possible drug with complex modifications assisted by PEG. And then even you are able to use it in the upscaling. So there's no decision from which point you need to switch or now have a complex PEG-made peptide, but then you need to switch to chromatography and come back to the problems you had before. So you can basically use it along the whole value chain.
Right. A couple of things that popped in my mind. I love the post-modification utility of it for especially saving on very expensive reagents, so different fluorophores and other modifiers, like you said. That's awesome, because then you're not trying to do the sort of the crude peptide being modified. It's just the one peptide that you're going to go forward with. Also, I know, I've read a publication of yours in the new antigen space. So maybe you could talk a little bit about how it fits in there.
Yeah, I can go. So in new antigens, of course, we have a very special situation, right? So you have a patient that needs a set of peptides very fast. So there is a sample taken, and there is some genome sequencing, then you get a prediction, you get a set of 20, nowadays also sometimes more than 20, like 100, depends, the peptides that are individually selected for this patient to trigger a T-cell response against the cancer. Now, these 20 peptides are unique for each patient, for each dose. That is, you need to manufacture them very quickly, but of course you administer it to patients, so you need to comply with the regulatory rules that needs to be applied, of course, because it's injected into a person. So you need to be fast and you need to get high quality, but you have every time a different set of peptides. So it's a very, very challenging situation. And as it takes sometimes years to develop one regulatory process for a single API, you need to do this in 4 weeks for a set of 20. So it's a very complex situation. But also here we see a huge benefit of our technology because, as Oliver already mentioned, we have kind of one size fits all, or one method fits all technology. So what you can do is you take those 20 peptides, you run a standardized synthesis procedure, and PEG has one nice feature as it chemoselectively isolates the peptide from its crude. Even if you have a bad synthesis, you will grab your product out of it. So if you start with a scale that's sufficient enough, you will always end up with your product in a purified form, and then you check on the purity. Sometimes with this catch and release methods, the purity will not be 95% right away. That's like the status quo right now. But then you can perform a subsequent polish. And what we learned from customers who already use this technology for neoantigen manufacturing is they would always use PEC as a first technology to purify and then run subsequent polish. But because you always get a product, you don't have to do resynthesis, saves you a lot of time. And second, this pre-purified product is typically much better soluble than a crude. So also the subsequent HPLC step is much more reliable. So our customers right now benefit from a reliability to deliver the peptide set in a given amount of time without having the risk, or at least with the minimized risk of resynthesis or of complex or issues during the solubilization step. So that's why the combination here in the neoantigen space between our PEC technology and classical chromatography is a perfect match to have a highly reliable manufacturing route, even for many peptides, every time different ones, in a very short time. We're now at, let's say, 3 to 4 weeks still, if we combine both, but our intention, of course, is to improve on our technology to be a standalone feature or a technology for purification. Then we'll really end up in a single day purification scenario, because it takes us 1 day basically to purify 20 to 24 peptides in different scales, or also on elevated scales. But yeah, right now there's a combination which is still beneficial, but in the future one hopefully does only need an automated PEC purification to be ready to go in 1 day.
So for your business model, I kind of asked about this before. You have the platform technology, the PEC, the peptide easy clean, but you've also started your own research programs. Can you talk a little bit about that?
Yes. So our business model is that, in principle, that everyone that works with peptides has access to our technology. Now we are coming, as a very small company, we are coming to a quite conservative market. So we are trying out different things actually to really get acceptance by the market, let's say. So we of course, we try to work together with companies, try to establish feasibility projects, integrations, and to really make aware that PEC is a viable option, that they can be trusted, to create a lot of data. And one way to do this is also to run a development project on our own. And this is actually what we are doing in a field where peptides find use, is the field of immunotherapy. And so this is where we were recognized by a very great program here in Germany to work on challenges in the context of viral diseases, where we use our PEC technology to make really great or potentially great drugs that have been proven to be manufactured very with many difficulties in the past. So this is our approach to go forward here as well.
Great. Are there also applications in the field of protein therapeutics? Have you started sort of thinking about expanding into that area?
Yeah.
Maybe you already have and I don't know about it.
Yeah, no. This is really really interesting field for us, of course. And also a field where linker-based methods are already more established in some way or another. I think our linker technology has been designed for solid phase peptide chemistries. So that's really how they are made. They withstand the TFA cleavage, can be cleaved later on by a very mild stimulus. So what we are currently doing is kind of in the hybrid field, where we are looking at peptide drug conjugates. This can be peptide antibody conjugates, peptide protein conjugates, and so on and so forth, because then the linker can be attached to a peptide, which can then further be attached and purified to whatever molecule you would like to, whether it be an antibody, a recombinant protein, or a small molecule, depends.
Great.
Maybe one thing that we, by ourselves, experience almost every day is that we started off with this linker as a purification linker. But in the end, we found the feature of the on-resin modification, for example. And we learned, and we had one project already in which we used the linker also to modify proteins. Because this concept of covalently capturing something on a solid phase after SPPS, and then having full control of doing several chemistries at this point, before cleaving the final product from the resin, offers many options. So the antibody drug conjugate field or protein modification field is one, but we also learned how we can do those different modifications. Now, for example, what we're exploring is also to run purifications in which we do not even precipitate after solid phase peptide synthesis, because we can directly take the crude and load it on the agarose. So there are many more possibilities to come with this linker, because it's robust. We have it, I think that's important information for some of the listeners. We can provide it in kilogram scales. So it's not an R&D tool. It's something that is made for industrial scale. Of course, we apply it everyone can use it. We have lots of academic customers who do very interesting and exciting things with the PEC linker. And it's also important to explore new ways of using it. But also, when it comes to the manufacturing of larger scales, it can be done with the linker as we have it in larger scales. So there are so many innovations coming out of this innovation already. And it's also one of the, let's say, fascinating things that for us, we're all scientists by training. Now we became entrepreneurs or founders and running the company, but still, I think it drives our enthusiasm to see also what we can do next with the technology. And there's so much more than just the purification. And that's maybe one of those possibilities that you mentioned with the protein modification, for example.
Yeah. Even thinking about something you said earlier of using the PEC technology and then HPLC as a polish, thinking back to the manufacturing scale where, you run a large column and you've got fractions, that sort of the hydrophilic and hydrophobic fractions on the side of the main peak that do have your peptide, that you can either combine those and re-purify them by HPLC, or you could maybe just take that big chunk that's mostly pure peptide and run that into PEC. So you could almost flip it on its head either way, right? To use the PEC technology prior or after. So if someone wants to try the technology in their lab, what's the easiest way? Just to go to your website? Do you have kits? Do you have where they could just sort of cheaply try it out to start?
Maybe one thing that, because we're asking for the business model. So the nice part is you basically need our PEC linker. But that's the core. Of course, we have the agarose, which is a modified agarose, and it's not, you can more or less, it's a custom-made resin, so you basically need two things. But the core is the PEC linker. So you can get the PEC linker from us in the form of kits. They're provided with reagents and consumables. You can get, let's say, manual handling, vacuum chambers, also from us. We can also just sell the PEC linker itself in terms of like supply agreements with customers who want to purchase it on a regular basis. And on top, we also offer, for people who do not have the in-house synthesis, we offer the services to run the PEC process ourselves and provide the customer with the PEC peptide, so to say, in order to give them the chance to make a first experience also if the peptide itself is adequate for their assay, for the drug development program. And then what happens then is, if it comes to larger scales, we have partners who can do the PEC process in larger scales. So the transition from R&D to larger scale is always given, either from us or then later from our partners.
Yeah, so I think that's really important to see that you could profit from our technology either if you're a peptide maker yourself or if you're a peptide user.
Yeah. So, I've asked you guys several questions. Do you have any, is there something that maybe I left out? We didn't really talk about limitations. There are a lot of really great things about PEC, but what do you find are some of the limitations, and are you working to overcome those?
Yeah, with every technology, there's always limitations. So if you ask me what one limitation is, that it's probably difficult to be a standalone method for API grade peptides or API grade peptides of, let's say, 99% purity plus, because in the manufacturing process there are always some impurities generated that cannot be removed by a catch and release based method per se. So this is for sure. However, I think we've come quite far to cope with this in order to identify also, let's say, very good tricks in order to avoid this these kind of reactions as good as possible. But there remains a limitation when it comes to final purity of the product, let's say.
Or may I add something here?
Yeah.
Because the status quo, however, for all API grades is you would basically never, you can basically not rely on a single purification run anyway.
That's right.
It's also a mind thing, because if you want to have 95, 99%, maybe in all of the cases you will have to do at least 2 chromatographic runs.
Yeah.
You always go for an orthogonal way. You start with, I don't know, ion exchange and then go for a reverse phase polish, or you have 2 reverse phase with different elements or gradients. So basically, it's not a problem per se. It's just here, too, we would couple our technology with chromatography. So instead of using 2 HPLCs, you can run with a truly orthogonal purification method and combine those 2 in order to get more reliable to your target purity, in fact. So, but yeah, I mean, that's something we also try to, convince the people in the market that it's the case. We showed a lot of times that we can achieve very high purities with this combination. We recently published an article about liraglutide, for example, manufacturing, in which we did this where we showed the PEC purification, at least as a, let's say, pre-purification before the HPLC polish can happen, for example. On other cases, we have shown that this works. But yeah, people are used to HPLC, but they will also use 2 HPLC. That's what's right. I wanted to add to Oliver's point, because 1 HPLC also won't do the 95, 99% in a single run. So that's the limitation that we share with other chromatographic methods. And then when you combine it, you actually have a better chance to get to your purity, even if you work with complex peptides, because then you can, again, take advantage of the chemoselective features.
Yeah. Maybe one other limitation that's is that our linker molecule is installed at the N-terminus of the peptide. So if you're working with N-terminally modified peptides, you can also install the linker maybe at a side chain of a lysine or so, but the maximal profit you'll get from the purification effect if the linker sits on the N-terminus. So that's maybe a limitation also.
Sure. Or an N-terminal lysine.
Then you have more options, yeah.
Right, yeah. Go ahead. Were you going to say something, Dom?
Nope. Nothing to add here.
I think something that, so I've spoken with like Rodney Lax and Lendert Vendenbos from NZTAG, and then Tina Bogle and Andrew Roller, where we talked about biocatalysis and RLS Bio's technology for developing tryptophan analogs is that the limitation, sort of to your point, is not so much the technology, but the outlook or the perception and the ability of a very conservative community to change what they're doing and the process and the way of thinking. So, it's a common thread. And I feel peptides have already faced that, like, their whole existence, right? Like, peptides have always sort of been this, like, no, really, we're great molecules. We can do drug-like. We can be in this space, and so and that's certainly been changing over the years, the perception of peptides, and so it can happen. Perceptions can change, So I'm hopeful for that. I asked you guys a question, and then I asked you another question. My first question was there something that you wanted to add to the conversation, something that I missed?
I think, yes, like peptides are a unique, is how to explain this. As peptides are a synthetic challenge by themselves. If you look into a drug development program with peptides, the people who manufacture the peptide will have an equally difficult and challenging time than the developers running their assays, right? Because if you look for RNA or so, you have basically, from a macroscopic point of view, or you basically have the same compound, you just change the sequence, right? So that's why you can purify oligonucleotides very rapidly, because you use the same method for all of them. To the machine, they look the same. But with peptides, you change one amino acid and the whole thing is becoming a nightmare. And this is what the community shares, right? So the people who work with peptides must be passionate because otherwise, they will get frustrated very fast. So it's part of the work to play with all the techniques available. And we consider our technology as an ideal tool here, because for all those things, the PEP technology or other technologies that emerge, also new green synthesis approaches, the trend towards those complex conjugates, everywhere we see a fit with our technology. It's like a glue that combines also different technologies and possibilities, like NCL on the solid phase. If you want to go for very, very long peptides, those complex modifications, combine proteins with peptides. Also, to just cope with the need for a high throughput manufacturing of peptides in a purified form. So at so many stages along the value chain, there are many, many challenges to be solved. And PEC won't be the solution for everything, but it will help to also combine those technologies and to work with the toolbox. And it's not an either or question if you work with PEC. It's like, use it and you open up a completely new portfolio of possibilities to work with the best possible drug. This is what we want to, as a value proposition, so to say, for the customers or for the peptide users, as Oliver said it. That they don't have to, make compromises in using not ideal peptide because it's otherwise not manufacturable. So it should always be the goal to work with the best possible drug as fast as possible. And we think that PEC is an ideal tool to do that for all the people, peptide makers and peptide users. Thank you, Oliver. I love that. 2 words.
Yeah, and in the end, I think it always boils down to 3 major things, which is doability or manufacturability, speed, and cost. And I think our technology can add value to all of these 3 critical points that people in the peptide field deal with.
Yeah, absolutely. I think that's something that I'm most excited about these days is all this technology that really allows people to be super innovative in their molecular design. So to your point that we don't have to have just the plain Jane, not that they're bad, but the natural amino acids and linear sequences that we can now look at a target and say, I can do whatever it takes to engage that target, because I'm going to be able to handle that molecule in the end. So that's what I'm really excited about these days. And I always like to ask people on the show sort of what you're, hopeful for and what you're excited for in the coming years. So maybe just, Oliver, you're actually on my screen right now as my speaker view. So I'll ask you first what you're most excited and hopeful for the next coming years.
So actually, I really share this passion of yours, because I think that's also one of the biggest differentiators that we as chemists can add in the field compared to the biological processes, recombinant peptides or proteins, is that we can increase or enhance pharmacological properties, stability, and you name it, by playing around on the peptide structure. And all these techniques like stapling peptides, fixing helical structures, or lipidating them to make them cell penetratable, or you name it, is really exciting. I read a lot of literature, what kind of uses these kind of peptides have, and our part can be that these molecules can be produced in a more easy way, maybe even in high throughput, stapled peptides or so on. So this gets me really excited to help in this aspect.
Dominik, how about you?
I think what makes me most excited is the development actually in the discovery area. So especially with artificial intelligence that's coming up, and which, especially in combination with peptides, provides such a rich pool of possible new drugs. And I think this is where we hook in with our technology already, right? Because this is always what I thought. There are so many leads that will come up by design, either from biological display technologies, or even from venom analysis, or from simply calculating, simulating using artificial intelligence. But if you end up with this rich pool of peptides, and then what to do next? And I always thought, okay, we must keep we need to make the throughput as big as possible to, make advantage of this huge potential, and not cut down the number of leads we follow because of some manufacturing issue. And the same thing counts for the complexity of the best possible drug. We should not, make compromises here. So that's why I think technology fits in very nicely, and I hope that we can show that also with our technology, there are more peptide drugs entering clinical stages and finally get approved later on. And that makes me excited to see that hopefully in the future.
Awesome. So something I did notice is that you both know quite a bit about the manufacturing process now. Is that from using different resources, or is that just from being in collaboration with manufacturing companies? Can you tell me how did you gain all of this knowledge?
Probably the latter, I would say, mainly. So getting involved into more and more projects. I mean, also along the development chain or value chain of the pharmaceutical industry, we were able at least to get, let's say, further from a lot of discovery projects, also to hit to lead projects. And so we gain more and more experience in the fields, and now even we have very good, let's say, we are working our way into the GMP space. That's the biggest hurdle, I would say, for us. But it's, I think, something that we are on a very good way to even enter this highly regulated space with our product. And so that's, we're learning by our customers, let's say, mainly. That's to answer your questions.
Okay, great.
Maybe it's important to say that, like, we do use PEG ourselves, and we provide also like peptide services, just to show how we can and what type of peptides we can do in which time scale. But our intention is to, if it comes to manufacturing, we want to make manufacturers be able to use PEG. So that's where it ultimately leads to, and those people have the most experience with manufacturing. Also, they need to comply with their rules, with their quality management. So it's difficult to, provide them, from our perspective, being more in the development innovation field, to provide them, this is how you're going to do it. Of course, we learn now, and we are dealing with the pre-requirements that we have to meet, right? So our linker needs to have a certain quality. We're making sure we have business continuity so that customers who start using our technology can use them for a long time, right? So it's not something that just happens to be there because we're a startup. And so we take care of all these things, and try to convince the manufacturers also that PEG is worth using also for standard peptides, because you save a lot of solvents. If you want to become green, PEG is one of the first options you should use in terms of downstream processes, right? So, but in the end, every manufacturer has its own specialty, its own scales in which they work, and their own, let's say, type of peptides they typically target. So there's always a part of customization of the PEG technology for the big API projects, but also we offer like services to create manufacturing process on the small scale that we already try to adapt scalability, look at the solvents, look at the economies that we have for each of those steps, and try to provide also reactors in the smaller scale that can be scaled up easily.
Yeah, and we don't have to do everything ourselves. That's a good point. I mean, we have our little space, that's the linker, the materials that we provide, and we can provide them in very high purity. But if it comes to the processes, the customers or the CDMOs that will ultimately carry out the project with our technology can really handle these, let's say, highly regulated processes.
And we've shown this, like this is already happening. So that's why we're pretty confident that this will happen much more often in the future. And then PEG won't be an innovation anymore. That's been one of the goals. It should be a new standard. Because then, peptide makers and users will profit. And that's, I think, that drives us right now.
Awesome. Perfect. All right. Well, is there anything else you want to add? Because otherwise, I think I'd love to end it on a very positive note that the PEG will become a standard.
I think
that's a good point to end the.
Good.
All right. Well, thank you both for your time, and all the resources you sent. The links, those will be in the description, so folks can follow up with all the resources that you have on the websites, your workshops, your webinars. All of that will be listed. So thank you.
Nice. Thank you very much.
Thank you for tuning in to this episode of Exploration Science. As always, we welcome your feedback and also your suggestion for topics that you'd like to see covered. If you enjoy this episode, please like and share with your community. Thanks again for tuning in.